My name is Raine. So I'm a PhD student at the Institute for Molecular Bioscience, which is a part of the University of Queensland here in Brisbane, Australia. And my project focuses on utilising blood feeding arthropods to see if we can try and identify coagulotoxins and also ion channel modulators in their saliva. So mammalian hemostasis is a mechanism that leads to the cessation of bleeding after we have some type of vascular trauma, and it's comprised of two different components. So we have primary hemostasis, which primarily involves platelets becoming activated where they bind to the site of injury and they form a primary platelet plug. This then leads into secondary hemostasis, which is also known as the coagulation cascade, and it involves activation of a series of different clotting factors, with the end goal being that there will be the conversion of fibrinogen into fibrin, which will then bind to the platelet plug and form a stable clot. So this system needs to be really highly regulated, and if we have any form of dysregulation, it can put individuals in a hypercoagulable state. And this means that they have an increased risk of thromboembolic events, such as thrombosis, where you have a big blood clot forming and then obviously vascular occlusion, or you can have an embolism where a piece of the blood clot will break off, forming an embolus and then become lodged in smaller blood vessels. And then this leads to different disorders such as pulmonary embolism, deep vein thrombosis, and also superficial vein thrombosis. So acute thrombotic events can be treated with short-term anticoagulant therapy, and this commonly involves heparin, so low molecular weight heparins, vitamin K antagonists such as warfarin, and then also direct oral anticoagulants, which will typically target factor 10A or also thrombin. However, although we have these drugs available, they do have limitations to them. So they have a variable dose response and also a narrow therapeutic window. So we want to try and identify new novel anticoagulants that can target specific coagulation factors. And the way we can do this is by looking at the saliva of hematophagous or blood-feeding animals. So we have a really wide range of different animals that feed on blood, both vertebrates and invertebrates. So some of the vertebrates you're probably familiar with are going to be things such as the vampire bats, the vampire finches, and then also the lampreys, which are a type of fish. However, the blood feeding habit is really dominated by the invertebrates and particularly arthropods. So we have over 14,000 species across 400 genera that actually feed on blood as a food source. And you're probably familiar with many of these, such as the ticks, the biting flies, kissing bugs, bed bugs, mosquitoes, and fleas. So the ones that I'm really interested in are ticks and tabanids. So within the ticks, we have approximately 900 species globally, and this is separated into 3 different families. So we have the soft ticks, which is also called Agassidae, and there's around 200 species. We then have the hard ticks known as Exotidae, which has around 700. And then there's a single species within Utilidae, which doesn't really have much medical or veterinary importance. So ticks are actually pool feeders. So they have piercing structures known as the hyperstoma chelicera that will actually pierce into the mammalian skin, causing tears, and then they can lap up the blood. And then they also have sensory structures called palps that sit on the outside. From the 900 species that we have globally, in Australia, there's 72, and of these, 17 have been reported to feed on humans and domestic animals. The most medically relevant tick we have is known as Ixodes holocyclus, and it's also called the eastern paralysis tick, and it's capable of causing death in dogs, sheep, and also potentially children. So with the tabanids, you may know these as either march flies, horse flies, clegs, or deer flies. So we have around 4,000 species globally, and it's separated into 3 subfamilies, which are then separated into 3 tribes within each of them. So within the Tabaninae, there's around 3,000 species. Chrysopsinae, there's around 600, and then Panganae has around 600 as well. The 2 that are in bold are the ones that are most medically relevant. Like the ticks, they are also pool feeders, but they have different structures that they use to feed with. So they have the labrum, mandibles, and lacinae. And the interesting thing with the tabanids is that only the females actually take blood meals, but this isn't all of the species, and it's because they require it for their egg development. In Australia, we have around 230 species, but unlike the ticks, we don't know any by species name that are medically relevant to the same extent. So the saliva of these animals obviously contains a really complex mixture of biologically active molecules, which will target the vertebrate hemostatic system to facilitate feeding. And they do this using things like vasodilators, platelet inhibitors, and also anticoagulants. So within the anticoagulants that have been identified in ticks, they will typically target thrombin, but they've also been found to target activated factor 10, the tenosate complex, and also other coagulation factor inhibitors. The most common class that is found within these inhibitors is a kinase type, and we have an example here of bufilin, which contains two kinase domains held like tandem kinase domains linked by a linker in the middle, and this is known as bufilin. Comparatively with the tabanids, not really much has been done on them. So there's only around 8 papers that look at the saliva and salivary glands of these biting flies. And they haven't really focused on anticoagulants, but rather all antithrombotics as a whole. So the first thing we ever found was tabanin, which was discovered in around the 1960s. Then in the late 90s to early 2000s, there was a couple papers that looked at the salivary gland extracts from around 25 different species of tabanids, and they found that they delayed clotting time and also inhibited thrombin. In around the mid-2000s, vasotab was isolated, which is a vasodilator, and this was found in the horsefly Hypermitra bimaculata, but they don't actually know how it causes vasodilation. And then most of the research that we know thus far has been from 2 different papers in around 2010 that looked at the Chinese species Tabanis yao, where they found multiple different anticoagulants, vasodilators, and fibrolytic molecules. So why do we actually care about this? So compared to other blood-feeding arthropods that are found globally, Australian species are vastly underrepresented, and this is because they don't really pose the same risk, because not a lot of our arthropods in Australia are vectors for anything. With what, as I previously mentioned, Ixodes holocyclus is definitely our most studied tick because it causes the most severe paralysis, but we haven't actually looked at the saliva or salivary glands of any other species that are native to Australia. And then this is also applicable to Australian tabanids, so we have no research done on their saliva at all. So Australian blood-feeding arthropods are a really good opportunity to try and identify new things that might not be found in other species. So this is basically just a summary of the workflow that I do for the data I'm going to be presenting. So initially, I'll start off by doing a proteotranscriptomic approach of both the saliva and salivary glands if it's feasible. And then once I've done this, I can go ahead and select different peptide and protein leads of interest. Once I've selected them, I obviously have to make them, and I can either do this using a recombinant E. coli expression system, or I can do it synthetically using solid phase peptide synthesis. Consequently, I'll then go on to purify them, and then I can test them on human plasma to see if they have any delay on clotting time. So to be able to do this, I have to obviously start by collecting animals. And so far, I've been able to collect 5 different species of exoded ticks with the assistance of Stephen Barker, who is a professor in parasitology here at UQ. Most of the ticks I've found so far, or have been given so far, have been from Ixodes holocyclus, but I have received several others, which are listed here. So basically, once I receive them, if they are in a viable state, I will attempt to collect saliva from them using pilocarpine, and that's represented over here. So basically, I'll attach the tick onto a microscope slide with tape, apply pilocarpine to its dorsum, and then see if it salivates. If it works, it will start doing something like this, where essentially you'll see the chelicera moving back and forth, and then hopefully saliva will come out. This isn't always successful though, so I have actually only been able to get saliva from Ixodes holocyclus and not any of the other Ixodes ticks, but I have been able to dissect salivary glands from them. With the ticks that are listed in red, these were actually used in an artificial tick feeding membrane system, which is demonstrated here. So essentially how it works is that you'll have a 6-well plate that's filled with blood. You then have a membrane that sits over the top. The ticks will then sit on top of the membrane and be able to pierce through and feed from the blood, which we attempted with this species. So I have been able to do some proteomics with the Ixodes, primarily the Ixodes holocyclus ticks. So the first ticks I received were unfed ticks that were ready to attach to their final host. And I dissected out the salivary glands because I wasn't able to collect any saliva from them, and did a reduced alkylated and trypsinide sample. And when I had a look at this, we had 43 different protein identifications, but they were primarily housekeeping and structural proteins. So this is basically consistent with the inability to collect any saliva, and it probably suggests that if a tick is not already feeding on a vertebrate host, it's not actively producing any saliva. So from here, I then did the same type of sample, but using salivary glands from ticks that had been feeding on domestic animals that were brought into local veterinary clinics. And this still needs to be annotated, but we found 400 different protein identifications, but from just having a brief look at it, it does still appear to be primarily housekeeping and structural proteins. So I did the same type of sample again, but instead now using saliva from the engorged ticks. And this still needs to be annotated as well. There were 65 different protein identifications, and just from doing like a preliminary analysis, it does appear that there's a higher proportion of secreted peptides and proteins. However, we haven't been able to find any of the proteins that cause paralysis, which are known as coagulotoxins, from having a look at it. And then finally, because I had the artificial tick feeding membrane system, we thought we could try and take the blood that they were obviously feeding from, spin it down, take the plasma, and then see if we could identify any tick proteins in that. But unfortunately, it didn't work. And this is likely because they are secreting their saliva into a really large volume of blood to begin with, and it's likely already also degrading at the same time. So based on these results so far, once I've obviously finished annotating this, it seems to be quite promising, and hopefully I'll be able to identify some anticoagulant proteins or peptides. And then I have also done proteomic salivary gland samples from the other ticks that I have listed, and we are just about to submit the RNA off for sequencing to construct transcriptomes, but this is still a work in progress. So I essentially do the same thing with the tabanids, but they're not provided to me. We do have to collect them from the field. So we can collect them using two different ways. Tabanids are pretty slow and docile. So typically if they land on you, you can just collect them directly with a falcon tube. Alternatively, we have something called a H-trap or horse trap, which is this thing here. So basically the flies are attracted to heat that radiates off this big black ball. They land on it thinking it's some type of host that they can feed from. And then when they realize it's not, their natural flight pattern is upwards. So they depart, fly into this funnel-like structure, and then obviously get stuck in the container at the top here. So we can use those two different methods to collect the tabanids. I've collected 5 different species so far, but we haven't been able to determine the species or subfamily for 3 of them. And because we collected quite low numbers of them, it's not really worth it. Because what we found so far is the salivary glands are very difficult to dissect from these flies to begin with. And actually then extracting or getting RNA is also a challenge again. So far we have been able to get saliva samples from Tabanus australicus and also do a thorax transcriptome on it. I also received a salivary gland transcriptome from another tabanid known as Pseudotabanus triferus, and this is one that's found in northern Queensland. And once I received it, I compared the sequences from the transcriptome to a database of coagulotoxic peptides and proteins that have been found in other blood-feeding tabanids for homology. And I did find 3 peptides, so PT5661, PT6991, and PT8082 that had similarity to the vasotab, which is a Cazal type peptide that was the vasodilator. And then there was 2 others, so PT6004 and PT7189 that had similarity to a coagulation inhibitors, which are kinase type, which have been found in the Chinese Tabanus yao species. So far, we've been able to successfully make 4 of these and purify them. And so I tested them a couple of days ago on plasma. So basically what's reflected here is the fold shift in clotting time. So anything that is above 0 is an anticoagulant, anything that is 0 is no effect, and anything below 0 is a procoagulant. And I found that one of the peptides, so PT8082, that has a similarity to vasotab, had about a 60% change in clotting or delay in clotting time compared to the control, which is really interesting, but this has to obviously be investigated further. So the next steps for this part of my project is I need to assemble the salivary gland transcriptome once I get it back for the 3 Ixodes ticks that I have. I also need to finish annotating the Ixodes holocyclus proteome, so both the saliva and salivary gland proteome. For the tabanids, I need to compare the proteomic samples I've run for the saliva of Tabanus australicus against the thorax reference transcriptome I've assembled. And I want to try and work out what the clotting factor that PT8082 is actually targeting within the coagulation cascade. I'm also going to then be doing EFIS experiments to try and characterize if any of these peptides are ion channel modulators and investigate that a little bit further. So obviously this project couldn't be done without the assistance of my supervisor team. So Dr. Andrew Walker is my primary supervisor. And then I have my other supervisors listed there. Steve has obviously supplied me with a lot of ticks. And then I've got the assistance from the King Lab members and also the Fry Lab members. So yeah, thank you for listening.
Now unmuted. So thank you very much, Raine. Are there any questions? Else I will start. I have first one that is not directly linked to you. Anyways, you showed also the fish and the bird. Are they also known to produce some saliva that support it, or are they just making huge wounds and fed then on the blood?
Yeah, so they would have, like any animal that basically is a blood feeder would have some type of like anti-hemostatic in it. It's just not all of them have been characterized so far. And typically the animals that are like longer blood feeders, like ticks that obviously stayed attached for a longer period of time, might have stronger anticoagulants, but not really many of them have been investigated that much, except for the really large vectors, like arthropod vectors. Like the kissing bugs are pretty heavily investigated in sort of certain species of ticks, but not so much of the other ones.
And the second question I have is, you showed that one toxin of the ticks are Kunitz type. Which is the size of the Kunitz? Because you showed there's a linker. So is it the double size of a Kunitz, or is it just one that is shaped like a bone with a normal?
So it's two Kunitz domains in tandem with a linker, but I don't know the exact weight of it because that's not my peptide. It's just a representation. Yeah.
So just a random one. Okay.
Yeah.